Organization and expression of IgG genes, diversity of immunoglobulins, structural variations in immunoglobulin constant regions. And this particular study note is developed by EPG Pachala team, MHRD. So I would like to express my sincere gratitude to the development team. The word immunoglobulin came at around 1930s when a class of proteins called globulins are termed as antibodies. Immunoglobulin or antibodies are the terms used interchangeably. Antibody is a Y-shaped divalent glycoprotein structure with heavy and light chain held together by disulfide bonds. On any microorganism mediated assault of host immune system, antibodies are generated against the antigen for its clearance. Antibodies are derived from mature B lymphocytes, WBCs of adaptive immune system, which on antigen binding becomes plasma cells producing clone antibodies specific to that antigen. In order to cope up with the enormous diversity of pathogens or antigens, antibodies require frequent adaptation to interact and compact against different emerging epitopes. So more than 10 billion varieties of antibodies are generated in humans, accounting for distinct sites on antigen for interaction. This body needs to generate this much diversity of proteins against distinct and diverse antigens. The immunoglobulin has very limited number of genes encoding for this. The range of antibody diversity outnumber the genes coding required to produce them. So, however, complex mechanisms are adopted by the human body to produce spectrum of antibody differing in their specifications or specificities and functions from that small set of antibody genes. By 1930, we all know that immunoglobulin belongs to globulin, a distinct class of proteins. Pauling and Pressman revealed that the antibodies have antigen binding sites called pocket where antigen binds non-covalently. Eisen and his colleagues suggested that divalent structure of antibodies. Campbell and Bullman in 1952 showed 700-degree angstrom limit of antibody site for antigen binding. Swedberg suggested the molecular weight of antibodies to be 160 kilodalton. Porter in 1950, with the help of a proteolytic enzyme known as papain, cleaved antibodies at the specific sites, resulting into 3 fragments, one with 50 kilodalton MW called the crystallizable Fc fragment and other 2 similar fragments with 45 kilodalton MW called Fab fragments antigen binding. Then Edelman and Ponlik suggested 2 basic subunits of immunoglobulin as heavy and light chain with 50 kilodalton and 20 kilodalton of molecular weight. In 1962, Edelman and Galli elucidated the nature of Bence-Jones proteins named after Dr. Henry Bence-Jones. If the patients with myeloma, they found dimers of immunoglobulin light chains freely secreted in urine samples and named them as Bence-Jones proteins. So later on in 1960, based on the carboxyl region sequence differences, immunoglobulin were classified into different classes and subclasses. With the development of X-ray crystallography, the antibody structures with this combining sites were elucidated in 1972. Among enormous and differentiated proteins found in the body, the most fascinating and critically important molecule is antibody, which provides protection against antigens for foreign molecules. The molecular structure of immunoglobulin is multifunctional with an additional advantage of gene duplication and rearrangement. Vertebrates can generate specific antigen binding immunoglobulin molecules varying from 10 raised to 10 or 10 raised to 4 or may be more in diversity. This unique characteristic of immunoglobulin Ig expressing structurally related still different molecules comes from the evolved cellular differentiation process of Ig producing cells where single immunoglobulin molecules specific for particular antigen is secreted from a single cell. The immunoglobulin is a globular structure with 3 partitioning. One is Fc region and 2 similar Fab segments with antigen binding region. The component of complement system as well as surface receptors on immune cells bind specifically to the Fc segment of immunoglobulin. Additionally, the movement of immunoglobulin across the membrane, placenta and body compartment are only possible because of these interactions. The movement of Immunoglobulin molecules at the site of inflammation is also because of such interactions. The best example of immune response elicited by this type of interaction is ADCC, antibody dependent cell mediated cytotoxicity, where the FC region of antigen bound antibody molecule is recognized by the innate immune cells such as natural killer NK cells and mediate a perforin dependent killing of target cells. Then antibody structure. 4 polypeptide chains combine together to form a pie-shaped divalent structure of antibody molecule. Polypeptides of 2 identical heavy chain of 50 kilodalton molecule weight comprising of nearly 450 amino acid residues and 2 identical light chains of 25 kilodalton of 214 amino acids are attached together by disulfide bonds to form a 4 polypeptide divalent Y-shaped structure. These disulfide bonds are critical for maintaining the divalent structure of antibody, and variations in the precise position of bond formation and number of bonds among different classes and subclasses of immunoglobulins suggest their role in the immunoglobulin structure. Antibodies of different specificities varies in the sequence and composition of heavy and light chain amino acid terminals within their first 110 amino acids. Therefore, the difference in the antibody specificity can be attributed to the diversity in the amino acid sequence of variable region called V region. The V regions are called VH in heavy chain and VL in light chain of immunoglobulin. CDRS or complementarity containing regions are highly variable regions in both heavy and light chain of immunoglobulin, forming the antigen binding site and traced to differences in antibody specificities. Here we could see variable region, disulfide bond, light chain, heavy chain, constant motion. Region beyond 110 amino acids variable region have relatively constant composition and same sequence in different antibodies and termed as constant region of heavy and light chain CH and CL. Immunoglobulin with some exceptions is glycoproteins with constant region restricted binding of carbohydrate. Although the exact function of glycosylation or carbohydrate attachment is not known, but immunoglobulin glycosylation enhance their solubility and via liver affects their clearance rate from the serum. Immunoglobulin fine structure. Protein organization at the primary, secondary, tertiary and quaternary levels determines the immunoglobulin structure. Amino acid sequence constitute the primary structure of both heavy and light chains. The primary structure forms polypeptide chains which folds upon itself to form beta pleated sheets. These extended chains form a compact globular domain as a tertiary structure. The continuous polypeptide chain of beta pleated sheets connects these globular domains. Finally, functional domains are formed as a quaternary structure composed of heavy and light chains, globular domains. It is this functional domain that acts as antigen binding sites and perform effector functions. Immunoglobulin fine structure. Beta pleated sheets held together with the help of disulfide bonds in variable and constant domains of immunoglobulin. Immunoglobulin domains. Amino acid sequence of heavy and light chains of immunoglobulin molecules comprised of repeated homologous units of 110 amino acid forming an independent globular motif called immunoglobulin domain. Within this globular motif called domain, a loop of 60 to 70 amino acids were formed by intra-strand disulfide linkage. Light chains of immunoglobulin comprised of variable domain denoted as VL and constant domain as CL. So, heavy chain of immunoglobulin structure comprised of single variable domain depending to the class of antibody and denoted as CH1, CH2, CH3 and CH4. Immunoglobulin domains folded into characteristic protein motifs forming a compact structure called immunoglobulin fold. Variable length of loops connects 3 or 4 antiparallel beta strands of polypeptides. These beta strands forms beta pleated sheets and are stabilized by hydrogen bonding. 2 beta pleated sheets form a sandwich structure to form immunoglobulin fold, and these beta sheets within fold structure are stabilized by conserved disulfide linkages and hydrophobic interactions. Although variable domains show slightly longer structure with extra beta strand pair and loop connecting this extra beta strands present in the beta sheet structure. Then variable region domain and CDRs. The variability among the amino acid sequence of heavy and light chain of variable domain includes exceptionally variable long polypeptide sequence termed as hypervariable regions. These short polypeptide residues are near 25 to 35, 50 to 55, and 95 to 100 amino acids. About 15 to 20% of variable domain structure comprised of such hypervariable regions, and 3 such regions are present in heavy and light chains of variable domain. The remaining stretches of polypeptide segment constituting variable domain is very much conserved in nature and termed as framework regions. The hypervariable forms the antigen binding site complementary to the structure of epitope present on the antigen and thus termed as CDRs or complementary determining regions. In the Fab, the 6 loops of hypervariable regions determine the variation of antibody specificities. Framework region functions as a scaffold that supports the 6 loops in place within the immunoglobulin structure. So CDRs of heavy chain of variable domain are non-specific and bind with high affinity to antigen than light chain CDRs due to the involvement of more amino acids residues of VH domain in antigen binding. Then hinge region, a proline, cysteine, lysine and aspartic acid rich extended polypeptide sequence between the first 2 regions of constant domain CH1 and CH2 in light heavy chain are termed as hinge region. It is present in the gamma, beta and alpha heavy chain, providing segmental flexibility. As a result, it allows FC and Fab segment to move relatively, thus acting as a tether to both the segments. Moreover, when antigen display is epitopes and FC move to bind with SA region, specific receptors on other immune cell surfaces to perform biologically effective functions. This hinge region provides flexibility because of which fragments of immunoglobulin can assume different angles relative to each other in order to bind with different epitopes and other immune cells. Proline at the hinge region make it more susceptible to proteolytic enzyme driven cleavage and also provide it extended polypeptide structure. This region is vulnerable to papain and pepsin cleavage. The 2 heavy chains are stick together due to intra-stranded disulfide bonds as a result of cysteine residues at the hinge region. Among different species and between different classes of immunoglobulin, the number of disulfide bonds varies at the hinge region. Although hinge region is absent in mu and epsilon chain of IgM and IgE immunoglobulin, a hinge-like additional domain lies between CH2 region of 110 amino acid residues in the gamma 3 and delta chain 60 amino acids are there in the hinge region, while alpha 1, alpha 2, gamma 1, gamma 2 and gamma 4 have 10 amino acid residues. Next is about diversity in immunoglobulin. Diversity due to domain variation. The chromosome has a relatively large segment called the loci or locus. Each loci for specific gene encodes for antibodies. Each domain of the antibody is encoded by several different and distinct genes. In humans, chromosome number 2 and 22 represent specific locus for light chain, kappa and lambda genes, while chromosome number 14 represent heavy chain genes. Each antibody structure consists of heavy and light chain. Both genes have variable and constant domains. Distinct B cells, not clones, generate different antibodies with much difference in variable domains. Then diversity due to hypermutation. The stimulated mature B cells rapidly differentiate and the genetic material of the cell also duplicates rapidly, because of which the genetic material incorporates point mutations or changes at very high rates. This process is called SHM or somatic hypermutation. Variable gene accumulate one nucleotide change per division and each SHM contribute to formation of broad range of diverse antibodies. Then diversity due to variable domain. 3 loops of hypervariable regions, namely HV1, HV2 and HV3 constitute the variable domain. Approximately 65 different genes located on specific locus of heavy chain encode for variable domains and they differ in their hypervariable regions. This set of genes, when undergo complex combination mechanisms, produce a large and diverse range of antibodies. This permutation, combination or rearrangement of genes is termed as VDJ recombination, as well as diverse range of antibodies are produced from different variable domains. Variable diversity and joining are segments of the genes which encodes for heavy and light chain of variable domain on the antibodies. Variation in the assembly of variable domain leads to structural variations in the immunoglobulin molecules. Diversity due to constant domain. Additional to all the above suggested variations, immunoglobulin structural variations called isotypes can be generated by linking some variable region to different constant domains of heavy chain. At the heavy chain locus, the genes for constant region are located downstream to the variable genes determining the isotype of immunoglobulin and effector functions. Based on the variation of constant domain of heavy chain, immunoglobulin is classified into 5 major isotypes. Secreted and membrane-bound forms of each isotype is produced by alternative splicing of mRNA of immunoglobulin. Isotype switching generates variable structural variations unique to constant domain of heavy chains, CH region. Then structural variations in constant region domain. Various biological effector functions are performed by immunoglobulin are determined by the amino acid sequence of constant domain. The CH1 and CH2 domains hold together the VH and VL domains of immunoglobulin via disulfide bonds and extend the Fab segment by virtue of the hinge region. Therefore, thereby facilitating the conformational rotation of two fragments and thus contributing to epitope binding also. Next part of this video will be uploaded by me later.